Good afternoon, everyone, and welcome to today's seminar in the ARC Training Center in Predictive Breeding monthly seminar series. Thank you for joining us today. We are pleased to have you with us for what should be a very interesting seminar on dairy cattle health, genomic prediction, and development of breeding values for trade that are difficult to record directly. So a brief introduction. My name is Seema Yadav, and I will be facilitating this session. I'm a research fellow at the Queensland Alliance for Agriculture and Food Innovation. So before we begin, I would like to acknowledge the traditional owner of the lands on which we meet today and the custodianship. We pay our respect to the ancestor and the descendant who continue cultural and spiritual connection to country. We recognize their valuable contribution to Australian and global society. So before I introduce our speaker, I will quickly go through a few housekeeping points. Today's seminar will run from 1 p.m. until 2 p.m. The seminar is being recorded, and the recording will be made available after the session through our seminar series webpage. Please submit your questions using the Q&A tab rather than the chat function. I will hold the question until the end of the presentation and read them out to our speaker during the question and answer session. So it's now my pleasure to introduce today's speaker, Dr. Amanda Chamberlain. Dr. Chamberlain completed her Bachelor of Agricultural Science with honors at the University of Melbourne. She completed then her PhD also at University of Melbourne under the supervision of Professor Michael Goddard. Her doctoral research focused on mapping QTLs for milk production, trade in dairy cattle, and using these markers in marker assistant selection. Following her PhD, Dr. Amanda joined what we now call Agriculture Victoria as a postdoctoral scientist. Since then, she contributed to several major advances in dairy genetics and genomics. Her work has included the implementation of genomic selection in the Australian dairy industry, contribution to the 1000 Bull Genomes Consortia and the functional annotation of animal genome consortia, and more recently, the Bovine Long Read Consortia. So today, Amanda will speak on the challenges of developing an Australian transition cow health breeding values. So with that, I warmly welcome Dr. Amanda Chamberlain. Amanda, over to you.
Great. Thanks, Seema, and thanks for that warm introduction. So firstly, I would like to begin by acknowledging the Wurundjeri people of the Kulin Nation, the traditional owners of the land on which I join you from today, and I pay my respects to their elders past and present. So firstly, the big challenge here is really for the cow. So transitioning from gestation to lactation is a very big challenge for dairy cows. Largely because of the reduction in feed intake and the very high energy demands of lactation. So this results in a cow that is in negative energy balance and has a reduced immunity postpartum. This has implications. It results in subclinical disease, infectious disease, metabolic disease, and subsequently poor fertility and reduced herd life. 75% of a dairy cow's health incidents occur in this transition period, so it's incredibly important. And in the words of Professor Jessica McCart, epidemiologist at Cornell University, these cows are running equivalent to 1.5 marathons every day in that transition period. So it's an incredible metabolic load that they are burdened with at that time. So the first challenge we had with this project is the recording of health itself. Many countries over the world have breeding values for health traits. For metabolic disorders like ketosis and displaced abomasum and metritis. In some countries, they have feet and claw infectious and non-infectious diseases. And mostly those breeding values are based on direct health records provided to genetic evaluation providers. They're often incorporated into health indexes as well. Unfortunately for us in Australia, health is poorly recorded, except for mastitis. This picture on the right is a data set that we were working with a couple of years back of 86,000 first parity cows. And you probably can't read the axes on the bottom, the x-axis, but you can clearly see that there's 2 bars that have reasonable frequency and the rest of them have very, very low frequency. The first bar there is mastitis, which I did say is reasonably recorded. The second bar there is actually missing definition. So there's very few other usable records for health traits in the Australian Central Data Repository. We looked at this for cows that were in parity 2 or greater, and the trend is exactly the same, if not worse. However, DataGene, who are the providers of genetic evaluations for the dairy industry in Australia, a couple of years back undertook their National Breeding Objective Review, and the results from that indicate that fertility and transition cow health are a priority for improvement. So that is the purpose of this project, is to build a health and fertility genomic breeding value. You've all seen this variations of this diagram before. To produce a genomic prediction, you need to build a reference population of animals that have the performance data as well as the genotypes. And so that's the challenge that we have. Coming back to this marathon running cow, we envisage a breeding value for transition cow health to indicate a dairy cow's propensity to successfully transition from gestation to lactation with minimal disease and successful subsequent conception and lactation. So the second challenge for us is what is the transition cow health phenotype that we're going to use to build this breeding value. Commonly, when we don't have direct records for a trait, we turn to intermediate phenotypes. And in health, this has been done before using somatic cell count for mastitis, but also calving interval for fertility. And in some countries, they've even introduced MIR, mid-infrared spectra, predicted biomarkers for subclinical ketosis. So in our project, we've used intermediate health phenotypes of biomarkers for metabolism, immunity, and inflammation to measure health during this transition period. This isn't plucked from thin air. So there's some previous work done at Agriculture Victoria by Dr. Tim Luke, where he looked at what are considered industry gold standard biomarkers for transition cow health, looked at the genetic parameters for it, and they evaluated their utility for genomic prediction. And so he estimated the heritability of some of these biomarkers, and they were low to moderately heritable. So certainly a good indication there that we can use them for genomic prediction. So what are they? So this is the biomarkers that we've included in our project. They sort of tackle various aspects of health. So the first panel there is looking at energy status, that's glucose, non-esterified fatty acid, beta-hydroxybutyrate, cholesterol, triglycerides. For protein metabolism, we're looking at total protein, albumin, globulin, the ratio of those two, and blood urea. For mineral status, we've looked at magnesium and calcium. Liver function can be indicated by bilirubin, but also cholesterol, albumin, and total protein. And we've got the inflammation biomarkers there of globulin, albumin, and the ratio. And we know that we can measure all of these in serum, and we've targeted our transition period to be from 1 to 70 days in milk. The third challenge is what size reference population do we need? And many of you will have seen this iconic diagram produced by Gonzalez Ricci et al. In 2014, which estimates the number of females that you require in a reference population given the heritability of the trait and the reliability that you would require. Given what we knew when we started this project, that the heritabilities of these biomarkers were low to moderate, but also that we plan to incorporate them in a multi-trait genomic prediction, we estimated that we could get a reliability somewhere of 30 to 40% with a reference population of about 10,000 and 10,000 females. So that was our target, I guess. So we collected, went out to collect blood samples from 10,000 animals with the aim of producing these health biomarker profiles for all of those individuals. The fourth challenge, which nobody likes to remember, but putting this presentation together, it certainly came to the forefront of my mind how challenging this was at that particular time. So a global pandemic is not a good timing to start a project of this scale. And I'm not sure if everybody remembers, but certainly those of us in Melbourne remember that we were one of the most locked down cities in the world. We were the most locked down city in the world, which meant we couldn't get out on farm to collect these samples for the first part of the project. The project started in July 2021, and 2 years into the project, we had collected 900 samples. So we were failing miserably, really, really struggling. And largely that was due to the consequences of lockdowns in the state of Victoria, us not being able to get out of metropolitan Melbourne, but also farmers having to deal with staff shortages because their staff couldn't attend the farm. They just lacked the resources to have us on farm. So it was really, really challenging time to try and put this project together. We did target the herds that were part of the Genomic Information Nucleus, which is an Australian reference population for dairy cattle genomic breeding values. So there's about 150 of those herds, and we were trying to target them for our project, but we were really not having much success at getting anybody in particular to come on board for the project. So enter some really amazing people that really turned this project around. We were really, really fortunate to encounter Mitch Crawford from Rochester Vets at a herd conference in Northern Victoria, and he just really, I think, valued the project and what we were trying to do, and he was amazing at recruiting herds in Northern Victoria. We also worked with Bernie Baxter, who's the lead supplier, large supplier engagement lead at Dairy Australia, and he just had access to many large dairy operations in Australia, and Laura Calder, who helped us vet all of these herds. So what we did was we offered commercial herds free genotyping, so effectively free genomics for the animals that we could take blood samples from. So this proved to be quite successful. Bernie sent out an email on a Friday afternoon to all dairy farms that had more than 400 cows in New South Wales, Victoria and Tasmania. And that afternoon, my inbox and my phone ran hot, and we had many inquiries into how to be a part of the project. So it was really, really amazing. So over the course of the next, I think it was 18 months to 2 years, we attended 17 different herds, collecting anywhere from 89 blood samples to the winner of the largest number of samples. We collected over 1,500. We actually had to attend that farm for 3 days, and they certainly were really helpful and really laid out the red carpet for us to be there to get that many animals sampled. We also developed a collaboration with Dairy Up. So Ian Lean and David Sheedy were really instrumental in putting that collaboration together, and they contributed 1,700 samples to the project from another project that they had where they were also collecting blood samples. So it was really great alignment of the two projects. And we have our Ellenbank Smart Farm down here in Victoria, and they routinely do these blood biomarker profiles for animals that are a part of their trials. So we were actually able to convince all of those project leaders to contribute the data from the Ellenbank herd that had been collected for various projects to the data set as well. So collaboration became an incredibly important part of the project. There's a group of people in that photo there that we had a big celebration when we finally ticked over our goal of 10,000 blood samples. There's a whole bunch of people that are not there that really, really contributed to getting this data set together. These are just some photos out on farm. It's logistically challenging to take these many blood samples, and like amazing team of people that we have at DairyBio, at Agriculture Victoria, that really coordinated all of this work. Amazing participating farms who just let us come. They let us set up our little mobile lab in the corner of their dairy, in their offices, in their kitchens. In one farm, they actually set up a marquee for us to set up in. But it really, really was such hard work, but a really, really enjoyable, great experience. I think everybody who went on these field trips found them really, really rewarding, even though they were a little exhausting. We even got the pleasure of taking the Agriculture Victoria's biosecurity van on a couple of visits, which was a little bit glamorous for us at particular times. But in the end, we collected over 11,000 blood samples. So massive, massive achievement. The fifth challenge here is, how do you validate a phenotype and a breeding value when you don't have those health records? So basically this little orange hexagon down here is the 10,000 Bloods project, which is our reference population, but it's just a part of a wider data set that we accumulated across the whole project. Hopefully you can see my cursor here. So the industry traits came from DataGene. They gave us access to the records from their central data repository for all of the traits that they routinely record. And then we had a couple of animal trials, this energy balance trial over here and the sick cows trial here. I'll give a little bit more detail of these and subsequent trials, but these were really multiomic projects that where we collected many, many different omics data, really targeting this transition cow health period. The first one obviously targeting the energy balance of the animals, and the second one, the I guess clinical disease aspect of transition cow health. And through the course of the project, a really, really valuable and important collaboration opportunity popped up, and I'll talk a little bit more about that in subsequent slides as well. So the first thing that most people ask is, well, are these biomarkers actually a good transition cow health phenotype? And I hope that I'm about to show you evidence that will convince you that they are. So using this 10,000 blood data set, Majid Kansafed has looked at the heritability and correlation of these particular biomarkers. And in this matrix, you can see the heritability on the diagonal axes and the genetic on the upper diagonal, and the phenotypic correlation on the lower diagonal. Basically, these are slightly different to the heritabilities previously estimated by Tim Luke. They're a bit lower, but they're still low to moderate heritability across the panel of the biomarkers. So we were pretty happy with that. And we confirm what Tim had also previously found, previously, is that they're largely phenotypically uncorrelated. So they're picking up very different aspects of the animal's, I guess, health status and their health and in this transition period, which is a good thing. So that means more of them is useful rather than fewer of them. Majid was also able to do a genome-wide association study using whole genome sequence genotypes for these animals. This is the Manhattan plot for beta-hydroxybutyrate. And you can see this really nice peak here on chromosome 23. And within that peak, there's a gene that's involved in butyrate metabolism and associated with metabolic pathways. So really nice sort of candidate gene that pops up in that region. And we found more of these types of associations as we looked through all of the biomarkers. Here's another Manhattan plot for cholesterol. Cholesterol peak here on chromosome 11 has actually a cluster of genes that look like they're involved in lipid and lipid metabolism. In particular, there's this gene ApoB, which is a known transporter of cholesterol involved in cholesterol homeostasis, and there's been a mutation within this gene reported that results in a cholesterol deficiency disorder in Holstein animals. So we were able to pick up this QTL, which was really nice to see. And we have many more of these interesting associations. The other thing we were able to do was to look at these animal trials that we had set up as a part of the larger project. So I'm coming back to this energy balance trial that I mentioned before, and this is work largely led by Sajid Tahir, and it's well known and well documented that negative energy balance contributes to this poor health, reduced production and poor fertility, as mentioned earlier. This is a really great diagram that Tim Luke put together when he was with us at Agriculture Victoria that demonstrates this problem for dairy cows in the transition period really well. So this is the race to conquer Milk Mountain. That is what he phrased it as, which I think is a really interesting way to look at this. But you've got all these cows that start their lactation. They start at the start line. The healthy cows, they are able to reach peak productivity, peak production, sail through their lactation with, having achieved peak performance. The subclinical cows, those cows in orange here, they start off, but they encounter some subclinical disease, which reduces their production, but they recover and they're able to at least successfully finish their lactation, though they wouldn't have reached the peak performance like we would have liked them to. And those cows that get clinical disease, they just really suffer, and they often actually exit the herd, which is really not an outcome that we want for our cows. So Shajib was able to what we did was we took blood samples and milk samples at 2 different time points. So we focused on that really early lactation stage where the cows are in extreme negative energy balance, and mid-lactation when the cows are returning to positive energy balance, but they're not yet pregnant. And we looked at the difference between the two. And if we just purely look at the biomarkers, Sajid actually has a whole lot more omics information, but just looking at these health biomarkers, 11 of the 13 that we're able to test are able to differentiate negative and positive energy balance. Hopefully you can see my mouse. So this is a volcano plot here on the left, and these biomarkers on the left of the volcano. It doesn't look very volcano-y because there's only 13 points, but the NEFA, BHB, and bilirubin are all elevated in negative energy balance, and that's what you would expect, knowing, I guess, the physiology of the animal when they're in that extreme negative energy balance. But you've got other biomarkers like cholesterol and triglycerides that are elevated in positive energy balance. And that again makes sense because they're back in positive energy balance and they'll have more circulating lipids available for their energy consumption. The plot here on the right, we plotted the first and second principal components of these samples, and you can clearly see that negative and positive energy balance stages can be differentiated utilising these biomarkers. The other animal trial that we ran, well, actually Anastasia ran, so this is Anastasia Kudryashova's PhD project. Absolutely amazing effort by Anastasia to successfully collect all of these samples. She's in the last year of her project, so she's trying to analyse all of her data. But basically, she was focusing on the comparison between clinically diseased cows and the healthy animals from the same herd. So she went out to a whole bunch of farms in southwest Victoria, in the region where she lives in Gippsland, and was able to collect samples from cows that were clinically diagnosed with ketosis, milk fever, mastitis, metritis, endometritis, and retained fetal membrane. She not only collected blood samples from those girls, but she collected samples from a matched healthy control from that herd, which had the same parity, same daisy milk and same production level. Subsequently, once she had the biomarker information, she was also able to determine which of the cows had subclinical hyperketonemia and hypocalcemia based on their BHB and calcium records as well. And for Anastasia, 12 of these 13 health biomarkers were able to distinguish sick from healthy controls. So she cumulatively put all the sick cows together and compared them with the healthy controls. She did this using 6 different methods. They're listed at the bottom of the graph down here. But this is the biomarker effect sizes from her limma differential expression analysis, comparing the diseased and healthy individuals for all of these biomarkers. The red bars here indicate the biomarkers that are higher in diseased animals, and the green in where they're lower in diseased animals. The grey one there is not significant. And the size of the dot just indicates the number of tests where she was able to confirm these biomarker effects. So really nice results that show that these biomarkers are certainly able to distinguish sick from healthy cows. She's also looked at the individual diseases. So again, we've got the diseases on the x-axis and the biomarkers on the y-axis here, and the dots indicate the significant biomarkers that can distinguish that particular clinical disease from its matched healthy controls. And so you can see she's got different biomarkers indicative of these different diseases. For ketosis, there's just 2 of the biomarkers that indicate ketotic individuals, but for retained fetal membrane, this one here in the middle, there's 7 biomarkers that look to be involved in that disease. She can distinguish sick from healthy animals with these biomarkers, but she can also go some way to predicting which disease they actually carry as well. Measured, coming back to our 10,000 bloods data set, looked at the phenotypic correlation between these health biomarkers on the x-axis and all of the industry traits predicted by data gene on the y-axis. So the blue boxes indicate those that are significantly correlated, positively correlated with the biomarkers, and the red boxes, those that are significantly negatively correlated. And just to highlight, I guess, here, if we're looking at some of these traits which we know would be related to transition cow health. So we've got somatic cell score, angularity, fertility, and 3 different survival traits. They all have correlations with these blood biomarkers. The other one that's interesting here, which is the third one from the top, fat yield, it also seems to have a lot of correlations with these biomarkers, which is not super surprising, given that a lot of these biomarkers also indicate, I guess, the lipid availability in the transition period, which can obviously translate into higher fat yield in milk. This brings us to this big collaboration opportunity that arose during the course of the project. And this happened off the back of a visit of Professors Jens Tettens, George Thaler, and Jorn Benowitz, all from German universities, that came across to Australia and Agriculture Victoria and met with Mike Goddard. And they clearly have a big interest in transition cow health and have their own projects. And so it was a really good opportunity to try and collaborate with them. Arising from that visit, we set up a visit for one of their recently graduated PhD students, Anna Maria Krzyniak. She came to visit us for a few months and brought with her this incredible data set for a whole lot of German Holstein animals that had phenotypes for these particular diseases listed in this table. So there's a couple of udder health diseases. I think there's 6 feet and claw diseases, metabolic diseases, and they also have a couple of body condition traits as well. And so they produce breeding values for these diseases in Germany. And so the project that we put together was for Anna to come and visit us and use Bayes R3 to estimate the SNP effects for these particular health traits in her German Holstein population. And you can see the numbers of cows she's got there, really, really impressive numbers. And she provided those SNP effects to Iona McLeod, who then used them to predict these German breeding values in Australian animals. Firstly, Iona did a bit of a sanity check because we have phenotypes for somatic cell count and mastitis. So just to double check that the correlation between the breeding values for somatic cell count and mastitis correlate with the Australian phenotypes. And you can see we've got really nice correlations there. Mastitis is a negative correlation, but that's just because the incidence is recorded in the opposite direction. So we're still really happy that the traits were similar in the 2 different countries. So Majid then took those German disease breeding values in our Holstein cows and correlated them with the biomarker breeding values that he had already predicted. Now, this is a table that shows the correlation between each of the individual biomarkers and each of the individual diseases or German breeding values. The correlations here are not high, but you wouldn't really expect them to be. There's a few little points there at the bottom when you're looking at the correlation between an Australian biomarker breeding value and a German disease breeding value. There's a lot of things to consider here. And one being the accuracy of the German disease breeding value, the genetic correlation between the disease and the biomarker in German animals, as well as the correlation between the German biomarker and biomarker in German population and the biomarker in Australian population, and the accuracy of our Australian biomarker breeding value. So you wouldn't expect these correlations to be high. So we were actually really impressed with the correlations that we did get. So in the red box there, you can see some really critical, correlations between these metabolic disorders and the biomarkers that we know are heavy indicators of negative energy balance. So we've got some really nice relationships there. The other thing that Sajid was able to do was to perform a multiple regression of the biomarkers and then look to see the correlation between the disease breeding values. And you can see on that bottom line that the correlations really do get quite high once you look at across all of the biomarkers combined. So this really boosted our morale, and we were really, really happy that we had access to a data set that could give us another way to try and validate these phenotypes. So this is just a summary to indicate. So across the top, we've got the what the biomarker does in response to energy balance, clinical disease, fertility, survival, and then these are the individual German traits as well. And you can see that largely the effect of the biomarkers is pretty consistent. So the top two here, BHB and NEFA, they're always bad, no matter which phenotype we're looking at. But then you've got others here that are just always good. So more cholesterol is good, more calcium, more magnesium is good. So largely these effects are fairly consistent no matter which, comparison we're trying to make with these biomarkers. So are our serum health biomarkers a good transition cow health phenotype? Well, we will argue yes. They're heritable and phenotypically uncorrelated. There is genetic variation associated with them. They're indicative of energy balance. They're predictive of clinical disease, and they're correlated with German disease breeding values, and combinations of them seem to be even more powerful than using any ones individually. So what would a transition cow health breeding value do? So we're coming back to our postpartum cow, and she's got all of these challenges ahead of her. But there are multiple things that affect an animal's fertility, survival, and their incidence of disease. And what we are proposing is that this transition cow health breeding value will improve the disease resistance, fertility, and survival that are compromised as a result of that negative energy balance and reduced immunity. So there are other factors that will still influence fertility, survival, and disease. And what we're really after is this center intersection of the three. And we think that we can make improvements to health by targeting that area. So Majid Kansavid was tasked with putting the breeding value together for our transition cow health phenotype. This is just a flowchart to show the different steps involved, and I'll go into each of these with a little bit more detail in following slides. But basically, the blue area is all performed within cows, and the orange area is all performed within bulls. So he started out by producing the breeding values for the 12 biomarkers, also produced breeding values for 7 industry health traits that we think are related to transition cow health. He produced the first principal component of those 7 health traits and demonstrated that they account for 50% of the variation. Used the 12 biomarkers to predict that principal component 1 for the 7 different health traits, and that formed the transition cow health index. Then backsolved to work out the SNP effects in the cow population for the transition cow health index, and used those to calculate the breeding value in a population of bulls. We then validated those breeding values with the 12 German disease breeding values and looked at the correlations between the transition cow health breeding value and the industry traits and indices that DataGene produce. So just to step through that with a little bit more detail. So to produce the breeding values, he used the 74,000 SNP genotypes that is routinely used by DataGene for industry evaluations. Did that in over just under 9,000 animals that had these 12 biomarker phenotypes and produced the genomic breeding values for the 12 different biomarkers individually. Looking at these were low to moderately correlated, as confirming our previous results, indicating that they're each probably picking up different biological pathways related to the cows. On the other side here, he took the those 7 industry health traits from DataGene, again using the same SNP genotyping panel in 65,000 Australian Holstein cows. The traits of interest were the first one here is actually exit 180, which is a breeding value that was developed by McConnon Hal Mariam here at Agriculture Victoria. And it indicates cows that exit or die within the first 180 days in milk. Then the remaining traits are all routine DataGene traits that are produced as a part of their evaluations. So cow survival, fertility, angularity, mastitis, somatic cell score, but we also included fat yield here because of that correlation with the biomarkers as well. A couple of these had to be multiplied by -1 to make sure they were all in a positive direction or beneficial direction. And the exit 180 is an EBV rather than GBV. But all of these traits showed strong correlation. And that strong correlation is evident when Majid actually predicted the principal components of these 7 different health traits. So you can see here that principal component 1 accounts for 50% of the variation in these traits. He also worked out the correlation between principal component 1 and the individual traits, and you can see they're all highly correlated except for fat yield. So this principal component 1 is really picking up these other traits rather than the impact on fat yield. He then predicted principal component 1 in a multiple regression model using 9 significant blood biomarker breeding values. So it turns out that BHB, NEFA, urea, calcium, globulin, and bilirubin all have a negative relationship with principal component 1, and magnesium, cholesterol, and glucose are positive impact. And so this is what we have termed our transition cow health index. All of these effects were significant at p less than 0.001, except for urea, which is significant at p less than 0.01. This is the correlation here between the principal component, the first principal component, and the serum biomarkers. You can see that they are correlated, but when you look at the correlation between the index, so our predicted principal component, the correlations certainly increase. So next, the transition cow health index was backsolved to calculate the SNP effects in the cows. This is the equation that he's used here, where H is the matrix of the genotypes of the cows and the G hat is the transition cow health index. Then with those SNP effects, multiplied them by the bull genotypes to get the transition cow health breeding values for this validation bull data set. So in this equation here, H is the matrix of the genotypes of the validation bulls, and the beta hat is the estimated SNP effects for the transition cow health index. And these the 7 health traits were all quite highly correlated with the transition cow health breeding value. So to try and validate this breeding value, our first port of call was to look at the German disease breeding values in about 8,000 of these Holstein bulls. So these are the same disease breeding values that I showed you before that affect udder health, feet and claw disorders, metabolic disorders, and body condition. And you can see from the final column there that the correlations are all in the direction that we would expect them to be. There is one exception here for white line disease, and we have a theory for why that has happened, but the good news is that the transition cow health breeding value certainly correlated with these German disease Majid then looked at the correlation between the transition cow health breeding value and all of the industry traits and indices that DataGene produce. So they're all listed there on the x-axis. The little boxes are around those that were actually used to produce the transition cow health breeding value and index. So we've got angularity, fat, fertility, mastitis, somatic cell count, survival in there, and the relationships there are all as we would expect. Cows that have a good transition cow health breeding value have good fertility, they have reduced incidence of mastitis, they have lower somatic cell count, and they have survival. They do have a negative correlation with fat yield, and as you would expect, because these are generally high producing cows, and the relationship with angularity is quite negative because angularity kind of reflects a dairy cow. It's a conformation trait, and that's negatively correlated with transition cow health, as you might expect. He then next looked at some of the industry selection indices that are produced for the Australian farmers. For those of you who are not familiar, there are 3 main indices for the dairy industry that are produced: the Balanced Performance Index, the Health Weighted Index, and the Sustainability Index. And I can see here on the x-axis the BPI, the Health Weighted Index, and the Sustainability Index are all positively correlated with transition cow health breeding value, which is really nice to see. There is another index, ASI, here, the second trait listed on the x-axis, that is negatively correlated with the transition cow health breeding value, but we expect that. That's an index basically of the production traits. And so high producing cows have poor transition cow health. So that's a really nice relationship to have picked up. The sustainability one was a little I guess, benefit that we weren't necessarily expecting. But if we move to some of the other traits, the first frowny face there is for dairy strength. So cows that are have good transition cow health often have poor dairy strength, and again that's a confirmation trait that indicates a cow's propensity for milk production, so that's not a surprise, as well as the other frowny face there, the milk breeding value because high milk producing cows tend to have poor transition cow health. The nice relationships here are indicated in the green boxes and the smiley faces. So we've got calving ease, a positive correlation with calving ease, as well as feed efficiency. But the one that we possibly weren't expecting is heat tolerance. I think that there is probably some more work to do to explore that relationship, but possibly what we are picking up is a real resilient phenotype with our transition cow health breeding values. So these cows are resilient to the metabolic challenge of negative energy balance, but they could also be resilient to climate pressures, environmental pressures as well. Finally, my conclusions. So producing a breeding value for a poorly recorded trait presented many challenges to us, but these health biomarkers did provide a good intermediate phenotype for transition cow health. The targeted trials provided biological support for biomarkers as intermediate traits, and our collaboration with some German friends provided a very important validation opportunity for this particular phenotype. So the transition cow health breeding values correlated with industry health-related traits, as well as those German disease breeding values. It's also positively correlated with the Balanced Performance Index, the Health Weighted Index, and the Sustainability Index, as well as potentially some other resilience type traits. And finally, well, not quite finally, but to acknowledge the team. I've listed names here that have been involved in the work that I've presented today, but there are more people that are part of the wider project team that haven't made it onto this slide. A really, really amazing team effort to pull this off. I think in a 5-year project, in particular, I need to shout out our participating dairy farms. The enthusiasm they had for the work and interest in what we were trying to do was really quite humbling. So I think I said to somebody before, this is a breeding value by the dairy farmers for the dairy farmers. They've really had a massive impact on putting this together. And we really couldn't have done this project with all of the collaborators listed there and acknowledged through the slides as well. The project was ambitious and these people certainly helped us bring the project to fruition. And finally, to acknowledge DairyBio funded the project, which is a joint collaboration between Agriculture Victoria, Dairy Australia and the Gardiner Foundation. And I'm happy to take questions, and I apologise if my connection has been a little poor.
Thank you very much, Amanda, for a wonderful presentation. And it's really a good overview of why TCH, transition cow health, is an important breeding target and how it's challenging to develop that breeding value when disease data is very limited. Okay, so now we have it time for questions. I have one question if I will ask from a very practical breeding scenario. So you said basically this health biomarker, serum health biomarker, they provide useful intermediate phenotypes, right, for TCH? For a breeding perspective, how do you see when you make a balance between this biological interpretability and prediction accuracy? When you need to decide which biomarker to include, does it make sense? Like you said.
Yeah. So we came from it, I guess, with some background information. So there was some previous work to look at these particular biomarkers and to test, to determine whether they were at least heritable and correlated with disease outcomes. So there was some preliminary work done in a previous project which gave the indication that they could be very useful. I think what's not surprised us, but certainly became obvious is that the biomarkers are all indicators of some quite different processes that are happening physiologically and biologically within the animal. And so they all capture their own, I guess, part of the transition cow health phenotype. So using them as a combination proved to be quite a strong, I think, phenotype. If we were trying to use smaller numbers or one or two of them at a time, I think that would have been much more difficult to demonstrate that they had impact. So, so there's that pre-existing biological information proved to be very valuable, I think.
Okay. Thank you very much. And we have another question from Ben Hayes. He's saying, wonderful work, Amanda and team. Could you use this info in a different way to inform farmers which heifers which they have already selected at a risk of needing help during transition? Can you get enough blood out of an ear punch so you could genotype and get a future performance prediction at the same time?
So I guess that's the idea with producing the transition cow health breeding value, is that could be done very early in life to predict the animals to retain in the herd that you think would transition better than others. We would be relying on the genomic prediction there rather than measuring the biomarkers within that tissue sample. So then I guess the question is the robustness of the reference population, and to make sure that we can maintain the reference population into the future to ensure that the accuracy of the prediction is still really good. So that's they're all questions that we have beyond this project. So this project wraps up at the end of June, and there's sort of questions that we have, and we'll have recommendations, I guess, to Dairy Australia and DataGene on how to progress into the future.
Thank you, Amanda. We have another question from you. This is from Suzanne. Nice work and overview of the great efforts by a large team. Well done. How are farmers using these breeding values, and how will you measure the impact at farm level or national level?
Yeah, that's a really good question. So it hasn't been implemented in industry yet. So these are the freshest possible results you could possibly imagine. I think Majid was still producing these last week. So we have met with DataGene to determine whether it's something that they would be willing to implement as it is. We would like to do further validation in other data sets to confirm the, I guess, relationship between the breeding value and some other ways of trying to measure animal health. But as it exists, I think we have been able to demonstrate that it does have utility as it is. So we are hopeful that potentially DataGene may implement this as a provisional breeding value, and we sort of can then start to survey farmers to see if they find it useful. I think that will be the way we have to progress into the future. I certainly know the farms that we went and collected the samples from were super keen for something like this. So there's at least a smattering of herds that I think will use it, but we'll need to, I guess, probably survey a little wider to see that other farms would find it useful as well.
Wonderful. Amanda, maybe we have one final question. I know we are already, it's two. It's a question from Owen. You showed a negative correlation between the TCH index breeding value and the expected breeding value. How do you see this trade-off playing out and influencing the adoption of the TCH-BV in selection decision?
Yeah, that's a good question. So the relationship between production and transition cow health is negative, basically, is what we have found. And this is not dissimilar to the relationship that we have with fertility, and we've known for quite some time. So in the dairy industry, selection was for productivity for a long time, and that resulted in very poor fertility within the Australian dairy herd. And that has been able to be reversed by farmers utilising both fertility and production breeding values to do their selection decisions. And we anticipate that this would be very similar. So it's a breeding value that would be considered, as well as the production breeding values, to balance improvement in production and improvement in transition cow health.
Thank you very much, Amanda, for sharing your work. Wonderful. And thank you again, everyone who joined the seminar. So before we close, I would like to let you know about our next seminar series. Our next session will be held on July 29th and will be presented by Professor Chakri Michel from University of Queensland. And we hope you can join us for that session also. And you can also view a recording of our previous seminars on our seminar series webpage, and sign up our newsletter to receive update about future seminars and central activities. So thank you again for joining us today. We hope you enjoy the rest of your afternoon. Thank you.